Hi everyone! Today, we will examine a novel imaging strategy for quantifying aging, as detailed in a recent Nature Aging technical report.
Here's what we'll cover. We'll start with the challenge of measuring aging and the limitations of our current tools. Then, we'll look at how a novel RNA binder was engineered to address this. We'll see how this system, called S-FLIM, was validated for detecting cellular senescence, and then how it was used to generate aging clocks in vivo. After that, we'll compare its advantages over existing methods, and finally, discuss the future of aging research in light of this technology.
Okay, so let's begin by addressing a fundamental challenge in geroscience, which is the objective assessment of dynamic aging processes inside living systems.
Our current approaches, things like DNA methylation clocks, are quite powerful, but they have inherent limitations. The process requires isolating DNA, which is destructive to the cells or tissue being studied. It's also time-consuming, and importantly, it only gives you a static snapshot at a single point in time. What's needed is an ideal clock that is non-invasive, non-destructive, and can provide real-time readouts, allowing for the dynamic monitoring of aging as it happens.
The researchers' central hypothesis is built on a known biological process. It starts with the idea that age-associated changes in nuclear RNA methylation alter the production of new ribosomal RNA. Specifically, it reduces the amount of pre-RNA. This shifts the ratio of mature to pre-RNA inside the nucleolus. This molecular shift, in turn, modifies a biophysical parameter, the nuclear micropolarity. And the key insight here is that this micropolarity is something that can potentially be measured with light.
So to test this hypothesis, a new molecular tool was needed. They required a specific dye that was sensitive to the micropolarity of the nucleolus.
The investigation focused on a class of dyes called hybrid polymethanes. They were engineered with two critical properties in mind. First, they needed high selectivity for ribosomal RNA. And second, their fluorescence lifetime, which is the very short time a molecule stays excited after being hit by light, had to be sensitive to the polarity of the surrounding environment.
After synthesizing several candidates, in vitro characterization identified FLP1 as the optimal probe. As you can see from the data, its fluorescence increased 410-fold upon bonding to rNRNA, which is a significant enhancement. It also showed a 3.23-fold higher selectivity for rNRNA over DNA. But most importantly, the last column shows its fluorescence lifetime ratio between nonpolar toluene and polar DMSO was 3.41, indicating the highest sensitivity to micropolarity among the candidates.
So, the conclusion was clear. FLP1 was the right tool. Its combination of high rRNA selectivity and a very sensitive fluorescence lifetime made it ideal. This lifetime property was shown to decrease from 2.7 nanoseconds down to 0.7 nanoseconds as the solvent polarity increased. This specific characteristic makes it a suitable probe for detecting those hypothesized biophysical changes within an aging nucleolus.
Alright, so with the probe selected, the next step was to validate whether it could actually detect a known biological state of aging, namely cellular senescence.
The mechanism here follows a direct causal chain. First, as a cell becomes senescent, its rnDNA methylation levels go up. This, in turn, reduces the transcription of new pre-rRNA. As a result, the ratio of mature rRNA to pre-rRNA inside the nucleolus increases. Because mature rRNA is more hydrophilic, this change raises the overall nucleolar micropolarity. And finally, in this higher polarity environment, the fluorescence lifetime of the FLP1 probe decreases, giving us a measurable optical signal.
This relationship was confirmed in vitro. As the paper states, the fluorescence lifetime of FLP1 decreases linearly as the mature/immature RNA ratio increases. The critical word here is linearly. This linear relationship means the probe's fluorescence lifetime is a reliable and quantitative proxy for that key molecular ratio.
Now, when this S-FLIM technique was applied to cells, it clearly distinguished between proliferating and senescent populations. The proliferating HeLa cells had a fluorescence lifetime greater than 3.0 nanoseconds. On the other hand, the senescent cells, which were induced with doxorubicin, showed a much shorter lifetime, less than 1.0 nanosecond. This corresponds directly to the underlying biology: lower methylation and a lower rRNA ratio in the young cells, and higher methylation and a higher ratio in the senescent ones.
So, having validated the method in cell culture, the research then moved to apply S-FLIM to generate aging clocks in whole organisms. The first in vivo model was the mouse. Neutrophils were isolated from mice ranging in age from 2 to 20 months, and the fluorescence lifetime of the FLP1 probe was measured in these cells.
A very strong linear correlation was observed. The fluorescence lifetime decreased from about 2.5 nanoseconds to 0.8 nanoseconds as age increased. This yielded a coefficient of determination, or R-squared, of 0.9699, which indicates a very tight correlation between the measured lifetime and the chronological age of the mice.
Following the mouse study, the same methodology was applied to neutrophils from human volunteers, this time covering an age range from 18 to 73 years.
And again, a similarly strong linear correlation was found between fluorescence lifetime and chronological age in the human samples, with an R-squared value of 0.9149.
A key feature of S-FLIM is that it's non-invasive. This allowed the researchers to track aging in real time within individual living C. elegans. They imaged the intestinal nucleoli of the same worms repeatedly over their entire lifespan.
The technique produced robust aging clocks for both the wild-type worm and a long-lived DAF2 mutant. You can see the R-squared values were very high for both. Importantly, the technique correctly identified the slower rate of aging in the mutant strain, validating its sensitivity to biological, not just chronological, aging.
Let's now directly compare the performance of S-FLM against some of the more established methods for measuring aging and senescence.
One of the most practical advantages is speed. A DNA methylation clock can take anywhere from several days to a week to get a result. The S-FLIM workflow, from sample preparation and staining to imaging and analysis, is completed in about an hour and a half.
And when compared to standard beta-galactosidase assays for senescence, S-FLIM also offers several advantages. Beta-gal assays are based on fluorescence intensity, which is semi-quantitative and can be affected by probe concentration. S-FLIM is based on fluorescence lifetime, which is inherently quantitative and independent of concentration. This also leads to much higher sensitivity. S-FLIM was able to identify senescence just 24 hours after drug treatment, a time point where the beta-gal assay could not.
Finally, let's consider the broader implications and some of the future applications for this technology in geroscience.
The capabilities of S-FLIM really open up several new avenues for research. Real-time analysis at single-cell resolution could allow for the creation of senescent cell atlases, mapping aging variation between different tissues. The speed makes it well-suited for high-throughput screening of potential anti-aging interventions. And from a clinical standpoint, it has significant translational potential for things like patient stratification or personalizing medicine.
This transition from static population-level snapshots to dynamic individual-level measurements of aging represents a significant advance for the field. It leads to the final question: how might this ability to get real-time quantitative aging data at single-cell resolution ultimately transform the way we develop preventative and personalized medicine?